Good afternoon, everyone. Thanks for joining in on LinkedIn Live for this particular session on clinical data science using R. We are delighted to introduce today's speaker, Dr. Priyanka Nair, a technology and talent strategist, AI researcher, and a corporate trainer with more than 11 years of experience in building enterprise-ready AI and data science capabilities. She holds a PhD in artificial intelligence, has multiple Scopus Index publications, and has led impactful AI ML, generative AI, and analytics programs across global organizations, delivering high impact training for industries including healthcare, retail, BFSI, and advanced analytics. Her deep expertise in AI, machine learning, and capability building, combined with her academic background as a former assistant professor, makes her a trusted educator who simplifies complex technologies into practical, industry-ready skills. We are honored to have her lead today's session on clinical data science using R. Thank you, Priyanka, for taking time out of your busy schedule and doing this for us. Over to you. You can get
Thank you
started.
so much for that introduction. So today I'll, take everyone here who is joining us live through the session, which is intense, high-level overview into a journey into the clinical research where it meets the modern data science. So I'll quickly share my screen. Can you please confirm if my slide is visible?
Yes, Priyanka,
Okay.
we can see the
So
slide.
I think trials today are becoming really complex, right? And the data sets are getting larger, and regulators are expecting cleaner, more reproducible and automated outputs in here, right? And that's where R truly shines in here. So this session, I'll take you through, a simulated demonstration of how clinical trial data can be, conveniently and in a powerful way be leveraged using R in here, be it data wrangling, be it statistical analysis, adverse event summaries, visualization, report automation, and ensuring compliance and reproducibility across the entire workflow. So quickly, getting into the start of this session. Before we get into directly into the demonstration, I really want you to look at the screen here, which talks about the clinical trial process and how seamlessly R can integrate at every phase of it. Although in SPSS has been, long used, in a long time. But then with its constraints, R has emerged to be that powerful programming tool, providing automations, be it statistical aspect. Now, starting with, if you look at the study design and the protocol development, right? So it starts long before the first patient is even enrolled, right? So R is being, just a moment. So R is being used by the statisticians and in the clinical research to simulate the sample sizes, visualize the design assumption, and support the protocol development and evidence-based decision making, right? Moving on to the data management and the cleaning part, I think that's where the very critical role of R, or rather any programming, would fall into place. It's really important that the data starts to flow from the EDC, right? The electronic data capture that we have. The focus has to shift to the data management, and it should be able to move its journey from EDC to SDTM, that is the study data tabulation model, and further it should be able to move to the ADAM, that is the analysis data model, and R is ensuring that, very, very effectively. Moving on to the safety and the efficacy profiling here, right? So, with this clean data, once we have cleaned the data, the teams can explore the safety and the efficacy trends in here. So, R allows us to really visualize the adverse events. It can be, detecting safety thresholds. It can be understanding how treatments can be performed across subgroups, right? So that's another aspect that can be effectively addressed by R in here. Decision making, very, very important, and something that's being effectively addressed by R and the teams, where the clinical science is actually meeting this strategy. Here you can really build those dashboards and simulation models, very, very easily in one click. It's not complex. And, with one or two clicks, you can build the entire dashboards once you have all the data prepared with you, right? So this is giving the operational teams, to have their finger on the pulse of the study, right? So I think decision making has been a really a key aspect with our going in lately. Moving on to the data collection and the trial, I think this is something after study we go to the data collection phase, of course. We have, this data, EDC data quality, we have to build the real-time dashboards as well. Once these prepared data, transformed data is there, we need to make some analysis, and basis those analysis we need to have some decision making, of course, out of it. These decision making aspects can be, further moved to the reporting and the regulatory submissions, wherein I think this is the most important and a differentiator to using R within clinical research in here. R is really empowering us to auto generate the TLFs, right? Be the table listing figures. It is compiling reproducible reports and streamline the regulatory deliverables. So, when I talk about the regulations, the regulatory aspects being addressed, it is well adapted and acknowledged by the FDAs, the EMAs, and the CDICs, right? So those are some of the a typical trial process, right from the design to data collection, to its cleaning, to transformation, analysis, profiling for safety, making those recommendations and taking those very, very important decisions, and then, of course, making it, submission, which is widely accepted by the regulatory on the regulatory norms, of course, right? So that's the entire overview of the how clinical trial process looks like and how R comes into picture. We already spoke about this, right? So why R accelerates this safe, reproducible clinical trials if we talk about, right? So in clinical trials, every decision, so we would all agree that how important or critical is it that these decisions that are being taken are to be accurate, it needs to be auditable, and it needs to be reproducible, right? And that's exactly why R is becoming the preferred choice in the pharma and the research. Now, if we talk about the reproducibility here, right? So, unlike the manual spreadsheet workflows, R uses the script driven analysis, right? Meaning, your results can be regenerated anytime with a single run, right? So this creates a reliable audit trail that regulators and the QA teams can really trust, right? Uh, we already talked about automation and reporting in here, right? So very, very effective tools, with R that comes into picture is the R Markdown and Quarto. It allows us to generate the tables, figures, narratives, and the full reports automatically here. So, this is really reducing the human error and dramatically accelerating the reporting cycle, honestly, here. Because, in this space, as much error can be reduced, I think that's the most meaningful thing that needs to be addressed while keeping the efficacy and the automation quality, in picture. Regulatory compliance, I think we have already spoke about that. These are the most acceptable outputs. The R-based outputs are acceptable by these standards. And in the industry, many organizations are already using R in their validated pipelines, making it really easier to create a transparent or maybe an auditable and submission ready workflows, right? And finally, I think cost and community is something that's a plus here. It's an open source, freely available, and it is continuously being enhanced by different communities, global scientific communities out there. So this is making it not only cost effective, but I would say incredibly adaptable to the evolving needs of the clinical research. So if you compare to all those solutions out there, R has been called out to be the best choice to be in the clinical research and the trial field, right? So these four are the key pillars in here. Now, when we look at the clinical trial data, right? Excuse me, it can seem really overwhelming at first. So, you can break down this scenario, right, of this entire, patient's journey, right, in just one piece at a time. So, you can look at it like each clinical data to be a shaped piece, right, it's a piece of the puzzle that you can call it. So, on its own, it doesn't tell the full story, of course, but then together it is creating a clear picture, for example, how well a drug is working, right? So the first piece could be patient demographics. It could be age, sex, and baseline characteristics, right? So this basically tells us who is in the trial and whether the groups are balanced, right? The next piece that you can talk about or think about is the laboratory results. It could be the chemistry, the hematology, all the numbers, right, behind how the body is responding. This is just like checking the engine light in a car, right? It shows what's happening on the inside. Then comes the outcomes and the endpoints. So it would include your efficacy, safety events, biomarkers, survival times, and more. So these pieces really reveal whether this treatment is truly making a difference or not. And finally, the visits and events, the scheduled appointments, procedures, vital signs, they're all providing a timeline, like the sequence of, building that puzzle or building a story, at the end. Now, once we have all these puzzle pieces together, right, we need to arrange them in a format that makes sense, that, can be put in a workflow and that can make sense, that can be transformed into some reports or dashboards helping us taking decisions. And those decisions, of course, can be, are ready for submissions, of course. Now, once we have all these together, uh, R really helps us transform the raw EDC. I've already spoken about this, that too. The standard is so EDC is the electronic data capture, the data that is captured directly from the sites, right? And it is converted into an SDTM data format. Now, which is again a standard format for clinical trials, for regulatory submissions, and then further converting it into ADAM data format, which is an analysis data format, right? So there are different tools that you can see here, not limited to something that you see on the screen. We'll also discuss, more, popular and, a wider base of these tools that can be used in here. And of course, the common tasks we have already talked about: importing, cleaning, transforming, analyzing, summarizing, visualizing, reporting, automating, and finally making it ready for submission. So this is how the analysis workflows goes through. Overwhelming at the beginning, you break it down into multiple pieces, feed them into the workflow, and you'll get your absolutely ready and limited error report ready with effective decision making out there. I think there are different use cases in the clinical research where it can be used together at a time, or it could be in stages that you can use for different submissions or milestones, I would say. So exploratory data analysis. So when we talk about clinical data science, it moves ahead in certain phases wherein exploratory data analysis becomes really important. Uh, it can generate the rapid, quick insights on the data that's there, right? In forms of the summaries, the plots. I think visually you can understand the data better, be it a bivariate analysis, univariate analysis, and to reveal really the patient data patterns, the hidden patterns in your data, the raw data that is converted into information. Safety signal detection, one has to compute the incidence rates, the trend plots, the spot unexpected adverse event spikes, right? So, what are the instances at which the AE, that is the adverse effect or the adverse event instances, rise there? Efficacy summaries, we have talked about, automated tables, figures, means, etc. Reporting and monitoring, again, interactive dashboards. I will be showing you, that in a bit, that how easy it becomes to visualize everything and track your study and provide your decision data points out there. Uh, Before we, jump into the use case, I really want you to, look at some of the very, very popularly adopted R libraries in the clinical research facet in here. Shiny, very largely used and very easy to use library out there in the R. It helps you build interactive web apps, right? So you can build your dashboards, you can build your trial monitoring facets out there, and exploratory analysis, right? So with Shiny, that interactive application based experience becomes enhanced. We have Plotly and we have ggplot, right? Both are used for visualization. However, Plotly, if you need more interactive visualizations like zooming, hovering while you hover on the data set, dynamic exploration if you want. So all these becomes easier with Plotly. ggplot is a more of a static visualization for publication and good quality graphs that you see. DPLYR is basically your main toolkit for filtering, grouping, summarizing, transforming the clinical data set. Of course, we cannot use it in the raw format, right? Uh, then talking about the TIDYR, again, a very intensely used library in here. It helps reshape the repeated measures, lab data, right? Essential for, adverse events, timelines, vitals, that kind of data about the patient, right? Radar, basically, it's used for importing the data in a faster way. Now, whether they are the CSVs or XPT or the large flat files that's out there, right? Knitr is, again, very much used with R Markdown. It is intensely used in the report formatting for the automated HTML or PDF tables. We'll see, how, this looks like as well. Again, Broom is another popular one. It's a tidy statistical results. It's majorly used in the reporting. Survival, again, it's time to event analysis for the safety endpoints. DT is for cleaner tables out there, right? Per is another library for functional use for automation, for functional programming, and scales out there. Since we have graphs, we have axis. So this axis and label formatting just to make your data more representable, more comprehensible, more interpretable. I think these libraries are used not just for visualization, of course. It is for the statistical analysis, like we spoke about, like TP LYR, or maybe TIDYR. It's mostly for the manipulation, summarizing, and the reshaping part. So this slide gives you an overview of some of the very popularly used R libraries out there. Okay, so let's look at one very interesting use case in here, which I'll simulate. Of course, we cannot go into the intricacies of the code at the moment, but then I really want to show you how this would make a difference, or how easily or conveniently your task can be automated, or you can report it, right? So just give me one moment. Okay. So let's say there's a sponsor who is running a trial with three treatment arms. Placebo means there is no actual treatments that's giving that's being done on the person, on the patient. There is a drug A that is being in the research, and the drug B. So these are the three treatment arms that we can look at. Now, as a clinical team, let's say you want to answer these aspects, right? How many subjects are enrolled per arm? What are the demographics? What adverse events are being reported? Are some treatment arms showing more side effects? Can we quickly generate a report for stakeholders? Can we look at different permutation and combinations of changing the values of these versus about the arms? So now these are some of the questions that as a team you would be required to answer. However, there are more aspects that might be required, of course, right? So in this use case, let's so these are the six things that we are trying to do. We are importing a clinical data set. Now here I have used a simulated data set, a synthetic data set, because again, there is no scope of the PHI here. So we have used a synthetic data set. We have prepared a synthetic data set here. Then we have wrangled the data wrangling part of it, the data analysis, summarizing, basically all kind of data manipulation has been done. Then we have, tried to join the demographics and the events. Now, demographics would be, like we called out the age, sex, and the site at which the data has been collected. And events would be the kind of effect somebody, is having an adverse effect, the spike in the adverse effect. Generate the summaries, automate the reporting, and we'll see how these are transformed. So quickly getting into the demo part of it. Please let me know if my art studio is visible.
Yes, yes,
Oh,
ma'am.
thank you. Thank you for confirming. Okay, now let's let's look at first. Just don't think about anything that you see on the screen right now. Let me just close these first, probably. Okay, let's look at our data that we have, right? So first look at the demographics data that we have. Let's import. So I am using RStudio here, as my IDE. You can use Visual Code, you can use Jupyter, irrespective of the platform or the IDE you are using. I am just, the intent here is to show the power of this language, or, rather the tool packages that comes along with are in here, right? Okay. So you can see here that, this is my demographics data. Of course, you will have lots of data in terms of the records. This is, like I said, that this is for the demo purpose. I just have 10 records of data. That includes a study ID that you see. Let me write on the screen. I'll just remove it. Okay. Okay. So here you can see. Okay, I'm not able to annotate. Okay. Fine. So study ID is, the study identifier, basically to study, basically the subject, ID that's there. Then we have the unique subject ID, that's again something that tracks the subject uniquely across all data sets, right? So this is specific to this data set. This is specific across all data set. Again, not going into the intricacies of this, let's look at, there is an age data point. What was the age at the time of enrollment into this, survey or this test? Sex of the subject, male or the female, the arm that we just spoke about, right? It's a placebo, it's a drug A, or it's a drug B. And there is a site ID that talks about, site where the subject was enrolled, basically at which site the test really happened. Now let's look at, the AE as well. That's like your adverse effect data sets in here. So again, the study ID remains same in here. So what you see here, code, it's like a med DRA coded AE terms, right? So that's like a standard dictionary term used for, not to get into the again the basics of this, but then just to know that we have taken a headache, fatigue, nausea, dizziness, right? So these are some of the safety stats that we have used. Then we have SDT. That's more to do with, when this adverse effect started, when this was reported. And then we have severity, whether that, the impact of this particular, safety stats was mild, moderate, severe. It is important for safety grading, right? Then we have, this is the flag that we have, whether this is a serious adverse effect. So for a specific problem, when somebody came, treatment was done, what was the adverse effect when it was tested versus while the test was done, or the impact of the medicine was taken, or the impact of the drug was taken in here, right? So AE is basically what side effects subjects experience, when and how severe. So this is basically showing the data that we are dealing with. Now I will go into the workflow here, right? Now, again, the time is too less to really, get into, the script, or, each nits and bits of this code. However, I would like to share that, what we are doing here. So we are loading the libraries. We just spoke about some of the libraries that makes it really helpful for different aspects of the clinical process that we spoke about, right? So we have, some libraries like, these imported. Then we are reading the inputs in here. I want to specifically talk about, so dm.csv and ae.csv. That's something that we already, spoke about. Now, there is another one that let me open. Now we have another one, dm.xpt. Now this xpt file is nothing but it's a SAS transport file, or you can also call as a SAS export format, which has this extension. So you can see that it has a similar data, but then just wanted to show that these kind of data records also can be handled with R, right? And XPT files are normally used for regulatory requirement, because FDA requires this SDTM and ADM data sets to be submitted as SAS transport, V5 XPT files basically. So these are logged immutable formats. So, but then for the purpose of demonstration, I'm using a CSV in here, right? Okay, now coming back to the demo workflow, our script that we were talking about. Here we are doing that data wrangling part, right? We are reading the input, we loaded the required libraries, we read the inputs, we wrangle the demographics that's there, basically grouping. It's like creating the groups, building a demographic summary table, let's say grouped by treatment arm, age group, etc. Right? So why this is being done? Demographics is the first table every clinical report really begins with. That's like the basic, right? And then what we are doing, we are joining adverse effect table. So we had two data sets, right? One is the demographics, and that captures the adverse effect that they reported at different sites in here, right? So we are joining the adverse effect to the demographics. So this combines the AE records with the subject level information from DM, right? So this produces AE terms, severity, subject demographics, etc. Right? Then we are creating a summary. Now, what does summary here means? Basically counting how many events occurred per treatment arm. So how many events of adverse effects happened with placebo, with drug A, with drug B. We'll see that, but then just a quick walkthrough of what we are doing with this code in here. Then we generate a AE plot. We see that adverse effect in here. We build a plot. We'll see that. And then we try to render a report. And then, so basically what the script is doing, this R script is demonstrating an end-to-end mini, replicated clinical workflow from raw EDC exports to clean data sets, summary table visualization, and finally an HTML report, right? So this is exactly how modern clinical programmers automate the safety reporting using R, right? So although this is a simulation, but then with in the actual environment also, this is precisely, the process that is followed in here. So let me run this code and tell you that how this looks like, right? So I've just run this R script in here, and you'll see that there is this plot that I have received in here. Now this basically shows AE count by the treatment arm. So you can see for drug A, for dizziness, the impact of how many were impacted by drug A, drug B, and when there was no drug that was provided for a certain safety standards, that was basically not safety standards, the problem that somebody came with, like nausea, dizziness, etc. Right? So this plot visualizes how many adverse event of each time occurred in each treatment arm. So be it, so the problem that was there, dizziness, fatigue, headache, nausea, across each of these AE term, what was the drug A, drug B, placebo count in terms of the adverse effect. So this was one of the outputs that we got. Now let's look at this summary that was created in here. This is demographic summary table. Okay, so you can see that this was again done while the wrangling, data wrangling that we consider. So this table summarizes the subject distribution by age group, treatment arm. So it's pretty easy to see the pattern that you can see for drug A, 50 to 65. The mean age is 60 in here, right? And N is the number of subjects in that treatment. You can, with this data, what insights you can draw that drug A is having a balanced distribution. Like one is less than 50, one is between 50 to 65. If you talk about drug B, mostly it's like 50 to 65 age patients, and placebo has older patients compared to the other arms, right? So they are having the adverse effect in here. So demographic influence can influence the safety results. That's pretty much that we are trying to show here. So for example, the older subject that is greater than 65 tends to have more adverse effects. That may explain, placebo, count in here. Let's look at other summary that it created for uh, adverse effect in here. So you can see that the arms you can see basically this is showing the frequency of each adverse effect term per treatment arm. So when I say AE term, I'm talking about dizziness or headache, nausea, etc. So what it is showing, it is showing the count of events for each of this AE code per arm. So it is like the underlying table used in the AE plot. This is basis which you got that result that I showed in the plot right now, right? So that's pretty much your R. So with this R script, we were able to do some minimal visualization, some statistical insights, derivation, so on, so forth. Now let's see, I want to show you one aspect of, I was talking about, dashboards, right? Wherein I am able to interact with my data, generate reports. So you can have the dashboard separately and report separately also. I've just created here for, an easy interpretation. So Shiny, like I spoke about in the previous slide, Shiny is very popularly used for app-based interactive app building. And this R Markdown is used for reports, automated reports. So let's see, playing this or running this data, what we'll get. So let's run this. So you can see that with a minimal code, you were able to create a minimalistic app here. And now you can see I can play around with the data. So let's say what I need to, you can select all these terms, right? So what kind of report that you want to generate. So I want to see the report of a drug, maybe a placebo, on all sites. Let me generate the report in here, right? So it's generated. Let me look at the report. Uh, So you can see it's an automated report that I was able to generate basis. I selected treatment as placebo, the site is all, and the subject analyzed were 5 in here. So this is the entire report that you can see here, and specifically the adverse effect plot by treatment: dizziness, fatigue, nausea, NA. For the so the arm specifically we selected placebo. Now just a minute. Okay, so let's say I put for all here, and maybe, so let me generate the report in here. So again, another report is generated just with a different aspect that I have created in here, right? So let me just, so this is what I selected for all treatment on site 1. what is the report that looks like, the adverse effect that looks like. It gives me the demographics, it gives me the adverse effect. So how you can see that how easily an automated report was generated, basis which you can make your insights, recommendation, and decision making, right? Okay. Separately, also, you can create an automated report using .rmd. I have used it R Markdown. Although the previous code that I showed you also had, an R Markdown, but then this is typically without an engaging, you can directly create a report as well. So here you can see that there is one aspect that we use, which is knit. So rather than running our markdown, we use a knit. So the moment you click on knit with the pieces that we have put in together, it will be creating a report for you. So you can see that there is a clinical demo report that has been created for you. So it is not an interactive one, but an automated report, right? So it can be customized, or rather it can be made as per the requirement of the scope of study and the organization that this study is running. So I want to show you one uh, part as well. Okay. I'll just show you one more, which is something that can be. So you can see that, here you have customized this, as per the requirement of the organization. And here, let's say, you can, play around with the age range. So this has been created again with the R Markdown. And you can, so let me say, I want to look at the efficacy at the end of week 4. So you can see how easily these, box plots have been created, showing, outliers or not. Then you have the pairwise t-test that is, shown at the end. Now you can also choose to play with different data to cross validate the correctness of the data that is being generated. So let's say, I regenerate the data, and you can see that these things keeps changing basis the data sample that I am working on. Again, let's say I want to look at the safety. So you can see that all these aspects can be noticed in here with respect to placebo, low dose, and high dose. I can choose to select one. I just, remove the high dose. If I, move the age gap, you'll see the kind of data moving very, very visual and easy to interpret and take decisions basis the requirement in here. And you can look at the trends for, let me add the high dose and things like that, right? So here we saw that, how we can wrangle the data, how we can summarize the data, how statistical analysis can be made with the data with different data sample at the same time. Also, we saw that high visualizations are possible in here. We can automate the report, we can have an interactive dashboards. So all these features come inherently with R, and it can be made powerful with as per the scope and drill down use cases. So one use case you can further drill down to different cases within that use case. So this, you can see the power of R within the clinical research, how much it can be leveraged in here. Quickly coming to uh, you know our deck here. Okay. So this is what we saw right now. So we have in our markdown template. So when you create a markdown file, you get a template. You need to add your requirements as per the data. You have, what kind of tables, what kind of figures. You want to embed figures, static figures, you go for ggplot. Of course, in when we talk about the Shiny apps, there we use Plotly, but then here, since it's static figures that we are talking about in the reports, it's ggplot that we have. We render the report, and these reports are good to be submitted as auditable outputs to the regulatory bodies, and these are appropriate for the regulatory documentation as well. So I think this is something that we already spoke about: the demographics. We standardize, we analyze, we do the exploration, and then we generate the TLF outputs. TLF is the table, listing, and the figures. Um, Now, I think R in clinical research, I strongly believe that it has addressed the manual clinical data analysis and the cost effectiveness that comes, cost addressal that comes with it, right? So, it becomes really clear that R isn't just a programming language, it is completely an ecosystem that we are talking about. An entire workflow can be addressed with this part, right? And if we talk about the manual clinical data analysis, of course, no doubt about it that it is inefficient, error prone, etc., right? We just talked about this automation part of it, which is so flawless. And I think this is pretty much what I love about this part. One click, and you can generate beautiful automations, and you can derive your insights in here, right? Exploratory safety signal detection, I think early safety signs, right? So, at the week one, week two, so you can flag it, right? The things that can become critical after, probably, a certain tenure of time. Then, of course, we talked about this. I can not reiterate enough that it creates a compliant data, and it makes you, take a data driven clinical decisions, right? uh, I think this, coming to the last part of the session today, it's really important that we stay ahead in the curve, right? So for somebody in the field of pharma, in the field of clinical research, I think these tools and expectations are evolving faster than ever, and it really means building a skill set that blends our AI community and the real world practice, right? So you can see that you can explore the Farmerverse tool, I would say, packages like Admiral, Turn. And these tools are really shaping the future of standardized clinical analysis. So I would really recommend to, explore these Farmerverse, right? If you talk about, practicing with public data sets, now I think PHI is something that comes along. So you can hit and trial, play around with the synthetic data before you actually start working on the real world clinical data. And there are a lot of public data sets also that you can get. So I think that's pretty much that you can try, before getting into the real world, into the real time data, of course. Automation, we have talked about connecting with the communities. I think there is a lot of people working in the research aspect of, how powerful R can be integrated with the with clinical research area. GenAI integration, although Python, comes with its own perks when it comes to AI and generative AI, I think it is the right time that you can upskill in the GenAI integration with the R markdown specifically. So I think AI is now part of model clinical pipeline. So from identifying outliers to generating synthetic data and exploratory analysis. So I think that's pretty much an area to explore and get into, as the pinnacles out there. ChatGPT plus RStudio, I think AI assistants, that are there like ChatGPT, integrated with R Studio can dramatically speed up the coding documentation and the report generation, right? So, whether you are exploring new tools, learning automation, or integrating AI, these are the skills that will keep you ahead of the curve. And I think becoming future ready in clinical research, is really the next thing in. So I take a pause at this moment and, would love to hear, if you have any, feel free to connect with me on LinkedIn or, send out your queries to the team. Happy to answer them. Over to you.
Thank you, Priyanka, for taking this session for us. And thank you all for joining today's session on clinical data science with our. A special thanks to Dr. Priyanka Nair again for delivering such a clear, insightful, and application-focused session. We hope the concepts shared today help you deepen your understanding of clinical analytics and the power of R in real-world healthcare data workflows. So we look forward to seeing you in the upcoming learning sessions. Until then, thank you and have a great day. Thank you, Priyanka,
Thank you
for
so much.
taking the
Thanks,
time
Sam,
out
so much for
from your
having me.
schedule. So always a pleasure. Good day. Good
Good
day.
day.
I'll stop the recording and end the session.